So welcome back, everyone. Let's get started with the second speaker. So our second speaker is Dr. Karla Addesso, again, one of my colleagues. So she is the Associate Professor of Entomology and a Center Director at Tennessee State University. And she's working at the Nursery Research Center in McMinnville in Tennessee. Her work focuses on sustainable pest management in woody ornamental production. She has conducted research on numerous pests relevant to nursery and landscape in the Southeast, including ambrosia beetles, flathead borers, Japanese beetles, scale insects, and mites. She is currently leading a nationwide project, which I am part of too, that to develop improved flathead borer management tools with a team of university scientists working in nursery, landscape, and orchard systems. So Karla, welcome, and the floor is yours, okay?
Okay. Let me share my screen. Hopefully it'll work the way it did the last time. Let me know when you see it.
Here we go. Looks good.
Okay. So thank you, Shamat, for the invitation and for allowing me to talk about this topic today. I'm going to be sharing kind of an overview of flat-headed borers and then gearing it toward our problems that we see in the Southeast. And before I start, for those of you who are not familiar with the Nursery Research Center, we're up in McMinnville, Tennessee. And so if you're ever up here and want to swing by, we welcome people to come by our research station. We're a little bit unique in that our research station only focuses on nursery crops and even more specifically, woody ornamentals. So we don't even kind of touching flowers and perennials is a little bit outside of our wheelhouse. So everyone here at the research center, including the faculty and some USDA scientists, all focus our efforts on woody ornamental production and the problems we see in nursery and landscape. And so you may have, and actually I think there are other scientists from our research center who have spoken on this series before. So you may hear from them in the future. I'm an entomologist by training. And so one of the challenges when I started moving into ornamental production research was I was used to working in vegetable crops or types of crops where the number of problems are limited. So, you know, if you're studying soybean or peppers, you've got your handful of diseases, your handful of insect pests, your production practices that are optimized. It's not as easy when we're dealing with the diversity of woody ornamentals. And so for our purposes and my purposes as an entomologist, what I'm trying to look for is focusing on major problems, things that are regulatory pests. So we do a lot of work on fire ants and Japanese beetles for that reason. Pests with wide host range so that spider mites love everything. So we can address issues that hit a wide range of problem plants, pests of high value crops. So if you have something such as a boxwood pest, it's going to attack something that is really valuable to the growers. And then also pests with few management options. So things that are challenging that we need to get a handle on in order to manage at all. So that's sort of our philosophy here on what we're going to focus on. So I'm going to be talking today about flat-headed borers, and there are a lot of flat-headed borers in North America. And so about 750 on this continent. They're known as flat-headed borers because of the larvae that have this sort of expanded front portion of their body, which is a combined head and thorax, and it's flattened. So it looks like a big flat head. The adults, though, can be very colorful and are often called jewel beetles, in particular because you can wear them as jewelry, and historically their wings have been. A lot of these are native to North America, and the ones that we tend to think of as problematic, such as emerald ash borer is probably the most famous and popular of the flat-headed borers right now. that one species did come from another continent and is an invasive species. But traditionally, these borers are found in forestry systems. They're part of the natural recycling program of nature, and so they don't usually cause too many problems. But that is not always the case. And so what I'm going to talk about today, sort of the focus of my research and my interest, is in this genus of flat-headed borers called Chrysobothris. And so for the rest of the talk, when I'm talking about borers or flat-headed borers, I'm referring to species within the genus Chrysobothris. Now, these are native beetles, so they didn't come from somewhere else. They've always been here, and there's a good diversity of them in North America. There's over 140 species. They're everywhere, from Canada all the way to Mexico, East Coast to West Coast. There are some species that have wider ranges and some that are just found, for instance, here in the Southeast. The most popular species that you hear of is flat-headed apple tree borer. And we're going to talk about why that name is a little bit problematic. But if you've heard of a flat-headed borer in this genus, it's probably that you've heard of flat-headed apple tree borer. And that is the species Chrysobothris femerata. And I'm going to tell you more about that. And actually, that's a picture of an adult and a larva of Chrysobothris femerata. So these beetles have, as I mentioned, there's a lot of different species, but they actually have a very typical life cycle that we can talk about. And for our Chrysobothris, the adults you'll start seeing emerge in May or June in Tennessee. And again, just thinking about where we are in the South, if I say something comes out in mid-May in Tennessee, it's probably coming out in early May in Georgia or further south. And if we're a little bit north, then you may be coming out a week or two later. So just kind of keep that in mind. A lot of the context of this is I'm going to be talking about Tennessee here in Middle Tennessee. So add or subtract a couple of weeks in your own area, depending on how you think plants are blooming in spring, if you're a couple weeks ahead of us. Mid-May, we start to see some of our beetles emerging. And then into June, they're mating. They start to lay eggs. And I'll talk a little bit about their eggs. Once they deposit their eggs on the trunks of the trees, the larvae hatch, and they burrow directly into the tree and through the bark surface, and then into and start feeding on the inside of the bark. And they spend their larval stages developing under the bark. They'll then burrow further in later in the year, in the fall, and create a little cavity and pupate in there. All of this early developmental stages, you're not going to see any damage. And that's part of the challenge and the problem of these beetles and managing them, is that everything looks fine until it doesn't. And so it doesn't look fine. Starting in October, if you're a good and careful watcher, you might be able to catch things earlier. But generally, I found by October, November, that's when we start to see bark breaking. You might start seeing some sunken areas. You might actually begin to notice that there's something wrong underneath the surface. And then through the winter months, with the temperature changes, that's usually if there's a cold or a warm, cold, warm cycle, you'll start seeing the bark break open and you'll see this powdery sawdust, which is their feces, called frass. And so it almost looks like a sawdust cake under there. And in some areas where you end up having sort of frost damage type situations, a lot of growers or landscapers, might mistake that type of bark breaking damage for frost damage. But if you peel back the bark, and you pick at the underneath of there, you'll see this sawdust cake come out. And that's how you can tell right away that it's not any type of physical, mechanical, or weather damage. It's an insect. And so they hunker down during the winter months, and then come April, May, they pupate to their adult stage, and they burrow their way out. And they often leave what we call exit holes. It's basically the shape of their body, like a little bullet coming out of bark. And so an old damage, you might see those exit holes and the bark might fall off. And again, for those of you who maybe deal with different types of wood-boring beetles, the flat-headed borers are really out on the outside of the tree trunk, feeding on the green tissue, the growing tissue of the bark, the phloem, and the cambium, and they don't really get into the heartwood or the sapwood. That's where you'll see more ambrosia beetle type of damage, where they'll burrow in and create little homes deeper in the branches or trunks. So if you're wondering what type of damage you're going to see from these types of borers, first of all, the most susceptible trees to these types of borers are newly transplanted and drought-stressed trees, and I'll talk more about that. Also, any trees that have been mechanically injured or grafted. One of the things we see a lot is that bud unions or areas that has grafting scars have occurred. It's a weaker part of the plant, and these beetles it's like buffet right there. They just are like, it's the steak, the filet mignon of the tree. That's where they're going. You'll often see damage kind of focused there. And as I mentioned, attacks are not obvious. If you're looking at trees and some of the symptoms beyond just the physical damage at the bark, is you might see things like suckering at the base. You might see, the thing to look at in August and September is if you've got a field of trees or a landscape where there's a couple of trees that are starting to turn fall color way before the other trees. That often happens with trees that have been attacked. And then you may see some sap leakage and trunk stains. This is kind of what I'm talking about as far as field. You could have crown death in nurseries. You can have, a field of nice looking green trees, and then some of them that already look like, they're going dormant a little bit early. And I already mentioned some of the damage, but these are just different degrees of damage that you might see at different times of the year. So one of the first things that people ask me when they think they have this type of damage is, what do I have? What beetle do I have? It's pretty easy to tell if we have a Chrysobothris borer. It's a lot more difficult to tell you which Chrysobothris borer you have. So I mentioned that we have this species, flat-headed apple tree borer. And if you look at the science and the literature and the history, everything is attributed to this one species of borer. As it turns out, there's a lot of different species that are closely related, and they're very hard to tell apart. And they're not just hard to tell apart for regular people. They're hard to tell apart for entomologists who are experts. There are only a handful of experts, and they all work on the team that Shamat and I are working on. And Shamat and I are not the experts because it's very hard. Like, there are people who have to dissect these guys. We've been working on some challenges of identifying these beetles. And the reason why we want to know which beetles they are is because insects are often very host specific. They come out at certain times. And so if we want to give good recommendations to people, we want to know, like, is it Chrysobothris femerata or is it Chrysobothris quadrimpressa? It turns out that quadrimpressa really likes to feed on oaks and femerata really likes to feed on maples. And so if you have one or the other, you don't need to worry about your other trees because the one species is only going to attack maples maybe, and the other one is going to attack something else. And so it's kind of important to know which species you have and what trees might be susceptible. We're trying to tease all of that apart to make better recommendations to people. And this is complicated because, as I mentioned, it's hard to identify these. The image that I'm showing you up there, those are 9 different species. They look the same to me without starting to dissect them. We've been working on different ways of identifying these beetles, and there's a couple different ways. Sort of our historical way of identifying beetles is morphological. You start You put them under a microscope, you start tearing them apart, you look at their legs, you look at their reproductive organs, you look at their heads, their antennae, everything, and see like, well, what's different between these beetles? We've also been looking at some other ways, other morphological ways to identify them. There's genetics, which is better, but also challenging, and I'll explain a little bit about that. And there's also some chemistry ways we can use to identify them. I'll talk a little bit about these different ways. Unfortunately, fortunately or unfortunately, I'm not really sure how to couch this. You can tell male beetles apart by dissecting their genitalia. Physically, the bodies of the beetles can look very similar, but once you start pulling out their insides, you can tell them apart. This is not something that just any person in a lab can do. It's not even something that any entomologist can do. And so it's not really practical as far as having somebody at a diagnostic lab just tell you what you have. But it's important because we get an idea of what the diversity is. The challenge is the females, a lot of the females look very similar or indistinguishable. So you can't tell the female beetles apart as easily as you can some of the males. So morphology is good, but it's not perfect, and it's not something that can be used by a wide range of identifiers just, at the state level. Again, these are some dissections and really highly close-up images. And again, you have to stare at hundreds of these before you start seeing differences. And, just as an example, when you're looking at Quadra impressa, like the left side is a little taller than the right side, and how tall is taller. And it's for people who are really obsessive about detail. This is a great line of work to get into, but it's not very quick and not very practical. So the answer to that is to turn to genetics and to see, like, can we do something a bit quicker that non-experts can use? And so we've been doing we have a team at University of Tennessee who's doing a lot of the genetics work, and they're trying to be able to tell the beetles apart using faster genetics tools. That is a little challenging with our beetles that are closely related to Femerata. And remember, I kind of said we've been calling everything flat-headed apple tree borer, but it seems like there are some beetles that are, maybe they're cousins, but they're not the same beetle. Well, the genetics is also very close in some respects. And it's possible that some of these beetles are interbreeding still. They may not be true species. And so we're still working on trying to figure out, is it one species? Is it 14 like we think it is? Are there more species that are just hidden in there? And so the genetics is helping. It's making it easier to identify some of these species, but there are a few groups of species that even the genetics doesn't seem to be able to consistently tell apart. And so that's been a bit of a challenge and makes it, again, as I mentioned, more complicated when we're trying to make recommendations. And if we want to tell people, spray for this beetle in, mid-May, but you've got the one that comes out in mid-June, we're giving the wrong recommendation in that respect. So we've been doing a lot of genetics. This is not my area of expertise, hence why the people at UT are doing it. And as I mentioned, some of the Femerata group we're kind of seeing some overlap with some of the species. We think that there are different species, but the genetics may be telling us maybe there's just a lot of variation. It's like saying if you just looked at a Chihuahua and a German Shepherd and you didn't know anything about genetics, you'd be like, oh, those are two completely different species. They might just be dogs with a lot of variation. We're trying to figure out, like, do we have a dog and a wolf, or do we have a Chihuahua and a Doberman? So that's where we're at with that. And we have about a year longer that we're going to continue working on this. And we're hopeful that we'll be able to at least get some answers as to whether some of these things that we're thinking are different species really may just be the same species. Now, one of the great things that have come out of this genetics work is that the larvae of the Chrysobothris, you can't tell them apart. They all just look like these kind of creamy little worms that are underneath the bark. And in order to identify them to adults, you have to raise them up to adult stage. So generally what we do is if we have an infested branch or something, we put it in a box, we wait for the larva to emerge as an adult, and then we identify the adult. And that takes a long time, and nobody has time for that. So we wanted to see, like, can we use the genetics tools that we've developed in order to identify the larvae so that we don't have to wait until it becomes an adult. And one of the challenges that we experienced in the past year is a very interesting one, in that some flat-headed borer larvae were found infesting pear fruit in California. Now, I just went, at the beginning of this talk and told you that these Chrysobothris feed under the bark of trees. And that is true. We've never seen a Chrysobothris in a fruit before. And so our first instinct was that, oh my gosh, is this an invasive species? Like, did something come over from somewhere else and arrive in California and start feeding on pear fruit? But we had some other colleagues in California who have been having problems with Chrysobothris in other orchard systems. And when they dissected and pulled the larvae out, they said, well, it just looks like a Chrysobothris, but I don't know why it would be in fruit. We pulled out the larvae and we did whatever the magic of genetics is, that some PCR, some DNA extraction, and looked at the DNA work that we had done to say, identify different species to see if we could match these larvae that were in the fruit with some DNA that we had already identified from species that we knew to be present in California. And it turned out that these larvae in the pear fruit are the other most common Chrysobothris that we have problems with, which is Pacific flat-headed borer, which is the more problematic species on the West Coast. It's kind of California, Oregon, and Arizona's flat-headed apple tree borer. They are related. And so we were happy that, one, we were able to confirm that this was not an invasive species from somewhere else. But we were also very surprised and also concerned that these beetles seem to be laying eggs in fruit and not in wood. Now, anybody who owns orchards or, has a fruit tree in their backyard kind of knows, like, when your apples or your pears start out, they're fibrous and more woody in texture and then they become more soft and moist and turn into something that is edible. And it seemed like these larvae, the beetles, may have started laying the eggs when the fruit were a little woodier. However, they might be seeking a level of moisture due to drought conditions in California and some other areas, that the fruit tissue just became more acceptable to them as a place to lay eggs. One of the follow-up things that we're looking at this year is, we didn't know if the beetles could actually develop to adults on the fruit, if that would be a dead end host is what we would call it. Like, they lay the eggs, the larvae tries to do something, but it never survives to adulthood. Well, it turns out they can. They harvested some of the fallen fruit and they just kind of let it dry out, in a bucket over winter. And sure enough, in the spring, they had adults come out of it. So in theory, these beetles could make a host shift from the bark and the branches into the fruit. And that is concerning, because if a beetle damages a branch or something on a plant, that you have to prune it, maybe you'll lose a little bit of yield. But if they're going to directly attack the fruit, now we have a fruit pest or a crop pest versus a landscape or ornamental pest. But that's sort of an example of why having some of these genetics tools can be so useful. Another thing we've explored is some chemistry work. And so one of the things that I learned very early on when working with insects is that insects and chemistry go hand in hand, because that's how insects communicate. And one of the things that we don't really think about, but is that insects have, and they secrete a lot of waxes and defensive compounds onto their bodies while they're alive. and the waxes are off for protection, or they allow water to kind of just roll off the back of them. And so we've been assessing essentially what a Chrysobothris smells like when you wipe the back of its wings. And would be something that when other insects are walking around and touching them, they'd be able to say, is this a mate or is this a different species and I need to find a different mate? So as it turns out, you can actually tell them apart more easily just by chemistry. And so this is these graphs are an example of the combination of chemical waxes that are on the surface of these different beetles. And I mentioned that females are really hard to tell apart. They physically kind of look much more similar. These are two species that are easier to tell apart. So we knew that these were two different species. And the waxes, the profiles of waxes on these females are very different. So even if you couldn't tell them apart under a microscope, if you just wiped the waxes off and analyzed them, you'd be able to tell them pretty differently. The challenge with this is that you can't really do this with museum specimens or old specimens. It has to be live beetles. But it's another tool we might have in order to separate some of the species that we can't currently do either with genetics or with morphology. The beetles, and actually I'll swing back to this, the external surface of the beetles, to me, is kind of boring looking. They can have green heads and faces. They can have sort of golden coloration on the bottom of their bodies. But under their wings can be a really bright bluish green color. And so one of the things we thought of is they have really big eyes, as you might be able to see. So if they usually when an insect has really large eyes, they are using them to see things. So one of the things we thought we might do is just some spectral analysis to be like, okay, we can only see certain colors really well, but maybe these beetles look really spectacular and maybe they look very different when you're looking at different wavelengths that human eyes just can't see. We've done some scans, and these are just different color scans of what the beetles look like. And so the ventral abdomen is the underside of their abdomen, and the dorsal abdomen is the top surface. And the ventral abdomen usually looks like a golden type of color, and the dorsal abdomen is usually this bluish green. But when you look at these three species, when I visually look at them, they look very similar. But when you scan them and you look at all the different wavelengths and what they look like essentially by a machine, what a machine sees, they actually look very different. And so this type of analysis can be done on museum specimens. And so even if it's not alive and it's been stored for a while, we've been looking at museum collections to see if these color identifications can be consistent, and especially between the ones that are just harder for us to morphologically tell apart. So we might have a way in the future through some of these methods, whether molecular or spectral, for non-experts to be able to identify them. So if you're going to scout for borers, IDing them is one thing, finding them is another. You're never going to find the eggs. And actually, when you're looking for adults, you're really not going to find the adults either. The eggs are these little tiny yellowish, flattened, little flat pieces of a circular flake, they almost look like. And they like to tuck them in under crevices or, as I mentioned, in wounds. If you can see the egg that female left on the right-hand side photo, and you can tell that from the bark at a distance, good luck. You're not gonna be able to do that. And the beetles, they're colored the way they are in part because it does help them camouflage into a lot of bark surfaces. And so you might be able to find them on wood piles and stuff, but you're not usually gonna see them on the trunks of trees. What we do have done and do use, at least to scout in farms, is using sticky traps. And as I mentioned, these beetles have pretty large eyes, and it seems like they can see colors. In particular, the ones that are here in the Southeast seem to be really responsive to the color purple. And if you have any recollection of, when emerald ash borer just started to show up in some areas, the APHIS USDA scouts were putting purple traps up in the trees on highways to try to see where those beetles are. And so a lot of these beetles, not just Chrysobothris, but other flat-headed borers, can also see in this purple region. And so we don't think they're actually necessarily seeing purple the way we see purple, but they can see in the blue region and the red region. And if you put blue and red together, it looks like purple to us. It might be that that's kind of what they're doing. And we've done a bunch of work with our problematic species, Chrysobothris femerata, and some others in the Southeast. And the thing that we've found is that the best trap for these beetles is a trap that mimics a tree trunk, a narrow tree trunk, which is odd because for many years we've been using these panel traps that we hang up in the trees, and that works great for emerald ash borer, but it doesn't work well for Chrysobothris. And when we first started some of our work, we were using the emerald ash borer trap, thinking, well, of course they'll fly to this square thing in the sky. But when you think about it, it doesn't really make sense for them to do that, especially because these beetles like to lay eggs on newly planted trees. They attack nursery plantings. So if they are using a visual cue, it's probably a small tree trunk or a small branch. And so we tried other types of traps, and they just didn't like the other types of traps. And it was much harder to catch them in them. We've been using these trunk mimics. And so it just makes it easier for us to track when the first activity starts during the season. It's not necessarily going to reduce the amount of attacks that you have in a field. We haven't shown that you can trap out these beetles, but it's a vast improvement for any type of surveying in an area if you're trying to figure out, when are we seeing activity start in order to make management decisions. Interestingly, the trunk mimics don't always work as well in large mature orchard systems. And we think that might have something to do with sunlight and shade. If you're in a pecan orchard or a walnut orchard, you've just got much bigger trees. There's much more shade, canopy shade. And so in nurseries, we kind of put these out as if they were a tree row out in an open canopy. And they work really well in the full sun. So we're still trying to figure out how to trap in sort of different systems with mature trees versus nursery production with smaller trees, but they work really well in nursery. For us, I've kind of mentioned mid-May is a good time where we start seeing emergence, but I wanted to throw some growing degree days in there for people in case you're, again, further south, further north, and want to have an idea of when. So we've been tracking emergence of the beetles from January 1. So with a growing degree day with a base temperature of 50 degrees Fahrenheit. And so what we're seeing here in Tennessee is that around the end of March, early April is when we're seeing pupae starting to appear. So the larvae are overwintering, but then they're molting into pupae around 238 to 389 growing degree days. And then our first adults, so they're still in the chamber inside the trunk, like they haven't come out yet, but they're there, is around May 6th, around 580 growing degree days. So they molt to adults, but they stay in that chamber for a little while. And part of it is they have to harden. So when you first see them, they're sort of this creamy white, just like the larvae, but then over time, they sclerotize, which is the external part of their body is hardening. And so they wait until that happens before crawling out. And so it's usually around mid-May. It's been May 11th, it's been May 15th, around 590, 600 growing degree days. So if you're concerned about, this is Chrysobothris femerata, flat-headed apple tree borer, the actual true flat-headed apple tree borer, that's around the time that you're going to see them start emerging. And some of the other species will come out a little bit earlier and a little bit later, but that's sort of our timing that seems to be pretty consistent in the Southeast. we've been collecting data. Shamat has been participating in this to try to figure out, okay, is that definitely consistent in different states in the Southeast? And can we just use a growing degree day calculation and instead of a calendar date, which is a little less reliable because we have cold springs, we have warm springs. And so May 11th is not always the same temperature as year to year. Interestingly, also from year to year is we find a lot of parasitized larvae, and I'll mention that a little bit later. So yeah, here are some of the species that we've had in the past couple of years, just to reiterate, kind of an earlier species and a lot of them coming out, and then some later species. And this is essentially the same type of data. In nursery production, we've been asked previously by growers, like, how small of a plant will they attack? And the answer is pretty small. Based on dissections of larvae in tree trunks from different species of trees that have been attacked, we're talking about trees that are about a 1/4 inch can be successfully infested. So very small trees, and maybe even smaller than that, they'll still lay eggs and try to develop in them. They may or may not be very successful, and there may not be enough food in a tiny tree like that, but it can damage the tree and kill it. So from our perspective, we don't really care whether they're going to survive or not, but they might have damaged the tree by laying eggs in it. Just something to think about if somebody asks you, well, what if we plant something smaller? What I will say is that because these beetles seem to be really targeting the newly transplanted trees for what should be kind of obvious reasons after listening to a talk on irrigation and keeping plants healthy, a smaller plant with a smaller, more equal root system that is being transplanted is going to get into the soil and establish much better than a much larger tree where you've maybe hacked off half of its root system and then transplanted it into a location. And so even though a smaller tree may still be capable of being attacked, it may not be, it may do better. So other things, if you're scouting, you may want to look on the southwest side of the trees. They like to lay their eggs on sunny side of the tree. The beetles need heat to develop. And you also want to watch herbicide drift, because we have found that herbicide drift does stimulate attacks. So we've tortured some plants to prove that, but you don't have to hit the foliage. Just hitting the trunks of the trees with like an overspray of Roundup, just accidental, when you're going around trunks, can cause attacks. And they like the smell of it. So we've shown that they can smell the difference between herbicided and non-herbicided trees. So, so be careful with your herbicide applications. Okay, so we're going to go into a few management practices, and I realize I talk slow, so I'm going to maybe speed up a little bit. Obviously, doing everything you can to do appropriate horticultural practices. And I'm going to really stress here irrigation for protecting against flat-headed borers. We've got more data over the past 4 years. We've been doing work, and the more we look, the more it appears that drought stress is a red sign, red light, come, food's here, and it's one of the reasons why you get attacks. So if you can manage as many of those horticultural practices to keep the plant happy, you're going to have less problems. Still, the best treatment we have for these is systemic drenches. Imidacloprid applied at transplant can give you, for a young tree, about 3 years of protection. We've tried other systemic drenches. They don't last as long, or they don't work as well as imidacloprid. I know imidacloprid has some issues. It's a neonicotinoid, and there's concerns about pollinators and beneficials, especially with flowering plants. So always read the label and follow the recommendations, especially if it's a flowering tree. But that product is still the best product we've tested. If you want information on application rates for nursery or landscape, this extension product has tables for the different formulations and how many mils or fluid ounces per tree and how many trees per acre you can treat. And it's updated regularly. So that is something that if you do need to treat with a systemic drench, it has the instructions for how to do that. And there's tables for the different types of formulations that you might be using. And we have found that on the label that half of the lowest rate is still effective. So the reason why this table has sort of the highest labeled rate, lowest labeled rate, and then half the low rate is because half the low rate works for these. So if you need to hit more plants per acre, that's the way to do it. We've tried some other of the newer systemic products, Acelepryn and Mainspring. They've been a little inconsistent. They're not as mobile, so they don't get picked up as well by the plants, which means that the health of the roots is very important to the effectiveness of the product. We've looked at Acelepryn and Mainspring, and so far, Mainspring is the one that appears at least to have some more consistency of being as good or almost as good as imidacloprid, but the rate is going to be really important, and how early in the season you apply it, and whether you're going to apply it as a drench or as a spray. And so for a lot of growers and a lot of landscapers, you might think about doing trunk sprays. The challenge with trunk sprays is that you're trying to either kill the females that are landing when they're laying eggs, or maybe with a bark penetrant, getting some contact product through the bark into the tissue so that when that larva hatches and chews its way in, it dies. But again, because there's so much diversity of beetles and timing, and they actually live pretty long, our beetles are living for like 40 days. They can lay a lot of eggs, and so you have to do repeat sprays. If you're looking for products that are labeled for flat-headed borer, I suggest you pick up the Southeastern U.S. Pest Control Guide if you don't have it. It's on the NC State Extension website. It'll probably be updated soon by the working group, and it has all the products, these tables with products that are labeled for flat-headed borers. And so that can give you an idea of what products are available and what products are labeled for flat-headed borers. It won't necessarily tell you which active ingredients are the best, but at least you know what you can use. We're currently repeating some tests, again, as I mentioned, with Mainspring as a trunk spray with a bark penetrant scrimmage. And it seems to be working about as well as the imidacloprid drench. I put a caveat on there is that, we're in our second year of testing, and there's also another product that is not commercially available yet. I'm hopeful that it will be at some point. And it also, in combination with scrimmage as a bark penetrant, seems to be working. Now, these sprays, though, in nursery were applied monthly. So mid-May, mid-June, mid-July. And again, for some growers who have major problems, or for some landscapers who, if somebody will pay you to do it, you can do it. But doing multiple, the reason why the imidacloprid drench is so adopted is because it's easy to do and it lasts a whole long time and you don't have to do repeat applications. But if there are people who are concerned about compound getting into nectar or pollen of flowers, these are maybe in the future, some other options. We've also just looked at other control methods. One of the old treatment wives' tales is just painting trunks with latex paint. That's not something you necessarily want to do with landscape trees, but in orchards, it does reduce attacks. It's not perfect. We've tried some other experimental products. We've tried Surround, the kale and clay product. They just wash off pretty easily and they're not very effective. But if you have orchard trees and the best you can do is paint their trunks with white latex paint, it might actually visually just confuse the beetles that they're looking for the wrong color They're looking for a tree trunk, and there's this white thing that doesn't look like a tree trunk. We've also found that, interestingly, weeds protect the trees from borers. So we did this insecticide treatment. I'm going to go back for a second. We did an insecticide and an herbicide treatment together. And we thought that, okay, if we keep the tree rows, the area around the trees really clean, the trees will grow faster, they'll take up more imidacloprid, they'll be more protected from the beetles, and it'll be great. What actually happened, as you can see here, if you look at just the 0 level, so that's no imidacloprid, the white bar is where there's no weeds, and the dark bar is where there's weeds, and the weeds essentially protected the trees from being attacked. And it was consistent for no matter how many rates of imidacloprid we used. So we used more and more imidacloprid, but the trees with weeds still had fewer beetles. And so that kind of led us to doing some work with cover crops to show that you can actually protect your trees just by growing foliage around them. In a production cycle, that's going to compete for resources, for water, but it also shades the trees and it keeps them from being ideal. I'm going to skip a little bit. It's going to keep them from being ideal habitats for the beetles. Of course, having cover crop around your production trees is going to reduce growth in those trees because they're competing. But if you don't want to do systemic drenches of 1,000 nursery trees, you can just grow a winter cover crop and allow it to grow up and naturally senesce during the summer. And that provides just as much protection as the imidacloprid. From a landscape perspective, one of the things that some of our landscaper collaborators are looking at is like, can they plant just companion plantings when they're doing landscape design, and to kind of shade or make the base of the trees not as attractive to the beetles. We've seen varying amounts of success with that. It depends on the type of plants that are being planted. Usually you're not planting fully grown plants around a tree when you're doing an original landscape, but I mean, in some instances you are. And so the size and height of the plants that are around the base, they need to be about 1 foot tall in order to shade the tree trunk and reduce the instances of the beetles getting in there. And then the big thing is irrigation. So we're doing some irrigation tests, and we did some work with cover cropping and irrigation to see like if we could use irrigation with the cover cropping to bump up the growth of the trees. And what we found most unexpectedly is that if we had no cover crop and we just irrigated our new transplants, we had basically very low numbers of beetles attacking. Irrigating in field situations is not a standard practice here in Tennessee. It just has not been historically needed, but we have a lot of late season droughts and periods where we just don't get enough water. And so if you can, being able to irrigate your new transplants will protect them. And it also sort of explains why we see a lot less problems of flat-headed borers in container production. Probably because you just have to be more diligent in maintaining enough water in containers for the plants to be successful. So by default, you're not creating a system where the beetles are particularly happy to be in. And then just some comments on using cover crops or being cautious and careful with your insecticide use is that there are natural enemies out there of flat-headed borers, mostly parasitic wasps. They're little tiny micro wasps, so you don't have to worry about them. They're not interested in you. They like to eat beetles, and you'll see them running along the trunks of trees, and they can hear and smell that there are beetles underneath the trunk, underneath the bark, and they pierce through there. They lay their eggs, and then these are little larvae of wasps that are eating the beetles underneath the bark. And so through some of our work, we found a couple species that have been reported before in other parts of the country that they'll feed on flat-headed apple tree borer. So these are 2. And then we've also found others that have not been identified, and we're in the process of identifying these. But I mean, they're out there, they're in the nurseries, they're going to be in the landscape. And so the more judicious we are with our insecticide use, we also have kind of in the background, these insects that are out there hunting for beetle larvae and controlling those populations. So we want to make sure we're not harming them while protecting our plants. In summary, we do have flat-headed borers that are native, impacting our landscape trees. A lot of these trees, the problems that we're seeing now are due to drought stress, and sometimes people growing plants where they shouldn't in areas where there's, I think the most common place you always see flat-headed borers is in a new parking lot where people stuck some trees, they didn't irrigate them, they just leave them there with a lot of asphalt, and you've just made a buffet for a flat-headed borer. So reducing that type of stress will protect from borers. There are chemical treatments that are available. There's pluses and minuses to the different active ingredients, to the different methods of applying them. And that altering the habitat around the tree trunks may be one option that is a non-chemical option for management, and also using irrigation, especially in that first year when the trees are establishing. And again, one last time, using spray products in particular judiciously to protect against harming natural enemies or other things and people that you don't want to harm. All of this work and more that I did not talk about has been funded by USDA NIFA and Southern SARE, with lots of help from different labs and people from even other institutions. And that QR code there takes you to a website that we're trying to update on whenever we get publications or extension materials that we put out on flat-headed borer. I will take questions, and that's my favorite bug. It's not real. It's a little Yoda as an insect. So I just like to end there.
Thanks, Karla.
Yep.
It was really good overview on flat-headed borer. There's no doubt about it. We are approaching 5 o'clock, so maybe there is time for one question. And I am looking at there is a question there already in the Q&A, and that's basically I'm going to read out. Do you see any availability of imidacloprid decreasing? It seems less readily available, maybe in North Georgia, I guess.
I have not. I mean, I know supply chains can be a little wonky at times, but there's no legislation or anything right now that's threatening the production or the access to imidacloprid. I would look for different label names or generics. If you're used to buying a certain product with a certain name, there are many because it's available generic now. I would just caution that you want to make sure that if you're used to applying a product with a certain rate, to always check the label, because the concentrations on those different products can be different. They can have other active ingredients combined with them. So you just got to be extra cautious if you're using something that you haven't used before. So I think that's all I really know right now.
Okay, looks like we are beyond 5 here.
Okay.
And thank you so much, Karla, for an excellent presentation on this, and really appreciate it. Okay.
Thank you so much. If anybody has questions, feel free to email me and I can share any of the more details, extension materials if needed.
Wonderful. Thank you so much.